Welcome to CSIR Institute of Minerals and Materials Technology, Bhubaneswar, Institute Lecture of Eminence, to be delivered by the pioneer in single particle cryo-electron microscopy and the winner of Nobel Prize in Chemistry 2017, Professor Joachim Frank. To begin the event, I invite the chief guest of the day, Nobel laureate Professor Joachim Frank, and our distinguished guest, Professor Bhanu P. Jaina, to the dais, please. And I request our respected director, Dr. Ramanush Narayan, Dr. Bibhu Ranjan Nayak, the Chief Scientist and Head of Mineralogy and Materials Chemistry Department, and Dr. Y.S. Chowdhury, Chief Scientist and Head, IMMT DSIR Common Research and Technology Development Hub, to accompany our eminent guests to the dais. Thank you, everyone. On behalf of the entire CSIR IMMT family, I extend a hearty welcome to our honorable guests and other dignitaries, invited guests, our mentors, directors from the neighboring institutes, colleagues, students, press and media, and each and everyone who are joining with us today for this Institute Lecture of Eminence. To begin with, I request our respected director to greet our distinguished guests with floral bouquets. Let us commence our program by invoking the blessings of the divine. I request our respected dignitaries to kindly join in lighting the auspicious lamp.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
Thank you, sir. Now I invite Dr. Bibhu Ranjan Nayak, Chief Scientist, CSIR IMMT, to deliver the welcome address.
Good afternoon. This is a proud privilege for me, for the Institute, and for the staff members of this CSIR Institute of Minerals and Materials Technology to have the most coveted Nobel laureate, Joachim Frank. We have Professor Bhanu P. Jaina. Welcome you, sir, for your time, and even we are sorry that the schedule is slightly delayed. And on the audience, we have Professor Wen Manti, we have Professor Hirendra Ghosh from Niger, Director, we have Professor Ganguly from Aijer Barampur, we have Professor Biswal, Professor Kaur, and many more, our senior colleagues. So, to avoid the formality, welcome you all in one sentence because we want to hear our two important lectures. One is lecture of eminence and one is a distinguished lecture. And I can also see Suvanja in the audience. She is from Imperial College London. So many dignitaries are there. So without wasting time, I welcome you all to this function. Thank you.
Thank you. May I now request our respected Director, Dr. Ramanush Narayan, to deliver his address to this august gathering.
जय जगन्नाथ, जय जगन्नाथ, जय जगन्नाथ. Today we are meeting at a historic moment of this institute 650 years of history. It is witnessed by so many distinguished persons who we would not have thought to bring at one at a time. But the credit goes to Jaina Trust. In such a short time, they have brought this much luminaries to this place. My due respect to our own Professor P.K. Jaina Pantri, our former director and Director General of CSIR, and Jaina Trust for bringing the people here. If I will talk in between two eminent lectures what is going to deliver, it will look like a salt. So I will not talk much, but only I will tell one thing in front of all of you, that today our Nobel laureate, the 2017, that only two correlations between both the person, both the friends, Professor B.K. Jaina and Joachim Frank, is that the care for the life of the people. One was experimenting with AFM in search of life, and the another person was looking through electron for life. And in Nobel laureate citation, if you see that this electron phenomenon, how it is going to help in the pharmaceutical and medicinal industry in the future. Science of today is medicine of tomorrow. Both the person from cell to electron, so they will be delivering, so I will not talk much between. But as a mark of respect to Professor Frank, Madam Frank is here, Madam Jaina is here, a hearty welcome to you. Thakur Sahib is there, Biswal Sahib is there, Hiren Ghosh is there, then our own Ganguly sir is there, Kar Sahib is there, Hirendranath Ghosh, everybody, I put a very, very hearty welcome on this occasion. And on all of your behalf and on behalf of the Institute, one of the lecture hall of the Institute will be named on Professor Frank. So when history should be depicted that it will go into the future, so there will be a lecture hall when Professor Frank, you will come again, you will see your name on our institution board. So this is our commitment from the Institute. So this is the third Nobel laureate visit to our Institute, and we have to pay respect that science brings always on the legacy, that whatever things has been created in the past and that will be done. So Hall of Fame, we are going to place Professor Frank, Professor Jaina, all the person, lot of planning is there in this progressive institute. I request all of your cooperation in this regard. This only these few words, because I am also habituated of talking very, very long, so I will not do that. So I will keep the stage for two of the magicians of world science, Professor B.K. Jaina and Professor Joachim Frank, to deliver their talk. Thank you. Jai Hind, Vande Uttam.
Thank you, sir. We are privileged to have distinguished Professor Bhanu P. Jaina from Wayne State University, United States, joining with us today. We have actually come across a short video describing his life and journey, and I would like all of you to kindly see the video. Right.
In 1996, a man discovers a new cellular structure, the porosome. Biology will never be the same again. This discovery rewrote our universal understanding of life on Earth. The nearly 38 trillion cells that make up each human being talk with each other through the porosome, the secretory portal of the cell. He hypothesized the presence of this structure decades before he saw it using the powerful atomic force microscope, and he did it alone. While others debated its presence, he focused on conducting the science that eventually allowed him to see and image the structure. That shattered previous dogmatic perceptions of the cell secretion that enables cell-cell communication and life processes, how the million miles of nerve cells communicate with each other for perception, thought, and memory, how digestive enzymes are secreted to digest food, how insulin is released to maintain glucose homeostasis, or how much is secreted in the lungs to lubricate and clear our airways were all revealed at the molecular level. This has allowed the treatment of diseases like cystic fibrosis and Alzheimer's and many more secretory diseases await its use. So what precisely is the cellular structure called the porosome? The porosome is a gateway or portal of the cell through which the cell releases chemicals, such as hormones or neurotransmitters, to the outside, thereby communicating with other cells in the body. It is also through the porosome that the cell releases digestive enzymes to digest food and antibodies to protect the body from pathogens. The portal is made up of some 30 proteins. Alteration of any of these proteins results in changes to other proteins within the porosome secretory machinery, resulting in diseases such as diabetes, Alzheimer's, and cystic fibrosis. It was previously unclear how the impairment of cell secretion could result in these diseases, since no one knew of the existence of the cell's portal, the porosome. The discovery of the porosome was thus a major breakthrough that is revolutionizing modern medicine. This is the story of one of the greatest contemporary cell biologists, who was called the father of nanocell biology in a scientific journal, and whose discovery has transformed both our understanding of cellular communications and the treatment of chronic disease. Dr. Bhanu Jaina was born on November 1st, 1955, in Jajpur, a small town near his ancestral village of Talhia in Odisha, India. He is the eldest of 4 children, born to Manju Prova Mohapatra and Professor Dr. Prafulla Kumar Jaina. His early childhood was spent in the remote villages of Odisha under the care of his grandparents, Dr. Braja Kishora Jaina and Tilatama Jaina. His grandfather, a devoted physician, chose to serve rural communities during both pre- and post-independent India, leaving a lasting impression on young Bhanu. These formative years, surrounded by nature and compassionate caregivers, instilled in him a deep sense of curiosity and empathy. At the age of 4, Dr. Bhanu Jaina began attending a village school, though he was more drawn to the wonders of nature than the classroom. Fascinated by mimosa plants and dragonflies, he found magic in the natural world, a fascination that would later shape his scientific pursuits. In 1960, following his father's return from doctoral studies at the University of British Columbia, Canada, the family relocated to Bombay, where Dr. Jaina began formal schooling. His mother, a gifted Odissi dancer, musician, and culinary artist, became his first mentor, nurturing his academic discipline and artistic sensibilities. Her influence fostered his lifelong love for science, music, and cooking. Dr. Jaina continued his education in Bombay and later in Banaras, Varanasi, where his father served as head and professor of metallurgical engineering at Banaras Hindu University. Summers were spent in Talhia, immersed in village life. fishing, foraging, and observing nature. These experiences, coupled with conversations about science with his father, laid the foundation for his future career. His first scientific experiment at age 10 involved observing sea sponges in a jar of seawater. The regenerative behavior of the sponge fragments sparked his interest in cellular communication and regeneration, themes that would later define his research. Inspired by the work of Nobel laureate professor Dr. Har Gobind Khurana, a close family friend, Dr. Bhanu Jaina pursued a career in cellular and molecular biology. He earned his Bachelor of Science from BJB College and his Master of Science from Utkal University in Bhubaneswar, Odisha in 1978. He began his academic career as a lecturer in the Department of Zoology at Utkal University. In 1979, Dr. Jaina married Meenakshi, his lifelong partner. Their shared commitment to academia led them to the United States in 1982, where Dr. Jaina pursued his doctoral studies in zoology at Iowa State University. Meenakshi completed her undergraduate degree in computer science and later earned an MBA from the same institution. Dr. Jaina's postdoctoral work at Yale University School of Medicine under Professor James D. Jameson introduced him to renowned scientists Professor George E. Palade and Professor Günter Blobel. These mentors profoundly influenced his scientific trajectory. A pivotal moment in Dr. Jaina's career came in 1996 with the discovery of the porosome, a cup-shaped structure at the cell plasma membrane responsible for secretion. Using atomic force microscopy, Dr. Jaina and his team visualized porosomes in live cells, revolutionizing the understanding of cellular secretion. Porosomes were found to be universal nanomachines composed of nearly 30 proteins, essential for the regulated release of cellular contents. His research demonstrated that mutations or disruptions in porosome proteins, such as CFTR in cystic fibrosis, or porosome proteins impacted by beta amyloid in Alzheimer's, impair cellular secretion. These findings have led to the development of therapeutic strategies targeting secretory defects in various diseases. Dr. Bhanu Jaina's contributions to cell biology have had a profound impact on human health and disease. His journey from a curious child in rural India to a pioneering scientist reflects a life dedicated to discovery, mentorship, and the betterment of humanity.
Sir, your journey truly motivates us.
Good evening. First of all, I would like to thank Dr. Ramanujan Nair, Director, CSR IMMT, and his organizing committee for inviting me to present this lecture. It is a great honor for me to speak at this occasion in recognition of my friend and colleague, Professor Joachim Frank, who has made two major pioneering contributions in science. Many of you may not know this, but he was the first to solve the structure of the entire ribosome many, many years ago, before people actually saw using crystallography. His second major contribution, as you'll hear from him, is participating in the development of cryo-electron microscopy that has completely revolutionized how structure can be gained for macromolecules, as well as not just the structure, but the dynamics using cryo-electron microscopy. So what I shall do today is present to you some of the work that we had done almost 35 years ago when we first identified the porosome. So I'll skim through it very quickly, but tell you its application. And at the end, I want to share with you what we are trying to do in not only translating biology, but also translating other aspects of development, such as food security, energy security, and health security. So this is a cartoon of a cell, as you can see, with many different components. And one of the major component is the porosome, which is at 5 degrees there. And it allows for secretory vesicles to dock, fuse, and release contents from the cell. So all of you had lunch, and your excrement pancreas are secreting digestive enzymes to digest your food. Once food is digested, your blood sugar goes up, and your beta cells secretes insulin to maintain homeostasis of the glucose metabolism. Similarly, you're listening to us, we're interacting, you're talking, and your brain's neurons are firing to understand, to remember, and to interact with each other. Now, the 2 pounds of brain that we have in our cerebrals, in our skull, if you line them up laterally, they are 1 million miles. So 1 million miles of neuron, which is equal to the shoreline of the entire planet. They're communicating, 1 million miles of neurons communicating with each other through the porosome, by secreting neurotransmitters. So just imagine the amount of information. Similarly, the 38 trillion cells we have in us, each cell has 6 feet of DNA. And if you line those DNA laterally, you'll have enough DNA that can go back and forth between the sun and the earth 61 times, which is 93 million miles each way. So huge amount of information processing goes on in a single cell in the whole body, and each cell is communicating with the other. So when this communication is impaired, it results in diseases. And that's what I'm going to show you in a moment. So first, let me take you through a little historical perspective. I did mention Professor Frank's work earlier, solving the structure of the ribosome. The ribosome was first discovered by George Emil Palade, who was my grand mentor, who won the 1974 Nobel Prize in Physiology of Medicine. And it used to be called the Palade bodies. And after that, Gunther Blobel, his student, solved how the cell's protein synthetic machinery allows for the new protein to be synthesized, to be targeted to specific subcellular organelles like the mitochondria, the ear, and so on. That is through signal peptide. And then in the 80s, Jim Rothman and Randy Shackman showed how these vesicles are derived from Golgi complex, and they're transported and packaged in membrane, so the contents are then transported to the plasma membrane of the cell, where they can dock, fuse, to release to the outside. And the final mechanism, And the final process in the secretory process is the docking and fusion of these vesicles at the base of the porosome complex, okay? And after we found this structure using atomic force microscopy, which is a force spectroscope, we were able to then, using conventional technique like electron microscopy or solution X-ray, we're able to determine the structure to be present. So you can see on the right-hand side, the top, showing you a cartoon of the vesicle docking at the base of the porosome, and the central panel on the right showing you a secretory vesicle docked at the base of the porosome for release. And so those structures were then identified and subsequently purified we were able to solve its, not only its structure, its dynamics, and the composition of the structure, both the protein and the lipid composition. In this EM micrograph, what I'm showing you is the apical end of the secretory cell, the exocrine pancreas that secretes digestive enzymes. And if you go to the top apical end, that's where the secretory vesicle docks and fuses. And it's shown by this little green box. And if you zoom in with an AFM, you can actually see very details in nanoscale structures at the plasma membrane, showing you pores that can dilate and in dynamics using AFM. So these studies were earlier done by a postdoc from Germany, Stefan Schneider, who imaged them with, initially we imaged them with handmade AFM and then later with digital instruments. And this is the surface of a live cell. And you can see that it is not flat like textbooks show, but rather many different protein complexes are there at the plasma membrane, ion channels, and so on and so forth. And if you look at in the little box, the circle that you see is a pit, and within the pit on the inset on the right, you can see the dark circles, 100 to 150 nanometer pores that are the porosome complexes. And this is just showing you by electron microscopy the structure that where a vesicle has docked, fused, and released its content in different scales. And you can see in D, actually, where we have outlined the structure of the porosome and the docked vesicle. You can isolate the porosome and reconstitute them into lipids. And you can see here, and on the left panel, you can see purified protein backbone of the porosome by negative electron microscopy and by hydrated AFM techniques. We can isolate the porosome and we can reconstitute them functionally in lipid bilayers. And in this, you can see in the EPC9 bilayer setup, if you have the porosome brushed into the bilayer, you can have secretory vesicles dock and fuse to release, and you can look at conductance and capacitance and measure amylase release from cis to the trans compartment. So biochemically show, indeed, there is functionality of the isolated porosome complexes. So those were pancreas, the very slow secretory cell. You can also do that in neurons, and this is what's showing you some of the nerve endings where the porosome is present, which is 10 times smaller than in the pancreas. This is about 15 nanometers in neurons. What is now interesting is, so you can, we know the chemistry, the proteomics. We also know, as I said earlier, using 3 different technologies, you can actually look at the porosome with the vesicle docked by AFM, EM, and solution X-ray. So very quickly, I got you through the structure. It took us about 25 years to get to this point, but I would like to now share with you how it could be translated. So what I'm trying to impress on you is that fundamental research needs to be funded, because unless you know how things work, and Professor Frank will present that, you will not know how to treat a disease when it goes wrong. Just like if you have a car, if you don't know the different parts of the car and how they're put together, then you don't know how to fix it when it gets wrong. So that's simple. Yeah. So the discovery that was done in the mid-90s, it took us 30 years to now fast track this, and finally we have one drug where we can take the porosome. So what happens is, if one protein within the 30 protein complex gets mutated or somehow misfolded, the rest of the protein within the complex are impaired in their function, and the entire nanomachine is impaired. So it's very difficult to just upregulate proper gene and have it corrected because it is a machinery, it's a machine complex. And so we finally thought that, hypothesized that if we take this complex, which is a nanoscale structure, for example, as I said, 15 nanometers is the size of a neuronal porosome complex. That means 7,000 of those can fit the thickness of a human hair. It's a nanostructure that can be reconstituted into a non-functional or disease cell and correct the defects of secretory defects. And that's what we were able to achieve in cystic fibrosis. We could actually not only treat the 2,500 different types of mutations in cystic fibrosis, delta 508 mutation is the most prevalent, but also people that do not have cystic CFTR gene product in their porosome complex, we can correct and treat the disease by reconstituting into the disease cell. And similarly for Alzheimer's, I'm going to show you data where we can actually do that and reverse the disease. So Alzheimer's, Dr. Alzheimer's identified that disease almost 125 years ago, and we still don't have, we have a management thing, but we don't have a reversal or a cure for the disease. And I'm going to show you data where now we are able to treat and reverse the disease. So in cystic fibrosis, the CFTR gene is mutated. It is a chloride channel. It's also a bicarbonate channel. And when that happens, it's part of the porosome complex, the CFTR protein. And when it's mutated, we found that nearly a dozen proteins are impacted on the porosome complex. And it impairs secretion of mucus. So all of you have breathe in dust and particles all day. And at the end of the day, everything is cleared. And the way it does it is because there are cilia that are beating in the airways epithelia that will clarify all the particles that are there through mucus, through the sputum. You can throw it out and get rid of all the particles, and whether it's bacteria or any other thing. But if the mucus is thick and viscous, then the cilia is unable to beat and clarify these. And that is a big problem because if they stagnate, then you get infections and there is disease, okay? So we found that, or people had found that there are a couple of those types of mucin 5B and mucin 5AC are lost in the mucus of patients. And when we checked that, we found that's indeed the case, but in the porosome proteins are impaired, not just the CFTR, but other proteins are impaired. That's the Western blot showing, and below is the cartoon giving you an idea that the hypothesis that if we insert a new porosome complex, we'll be able to overcome the disease. So in this, I want to take you through very quickly on this. What you're seeing is porosomes that have been added to. So the green, some of the green fluorescence that you see are porosome complexes. So in CFTR, you don't see them in the lungs very well. If you reconstitute them, then you can see them on the plasma membrane, okay? That's what essentially it's showing you. Okay, so if you look at the top panel, you can see the control PBS. Then you have added 100 nanograms and 200 nanograms of the porosome in day one into the human airways epithelia organoid. That's what FDA wants, organoids, human organoids over animal studies, but we have done animal studies too. And if you see that even 100 nanogram of porosome is equivalent to, in the day one, Trikafta. Trikafta is the most recent drug by Vertex Pharmaceuticals, and Trikafta treats only some of the patients, about 12%, 12 to 13% of patients, not all patients. and its annual revenue is $12 billion a year. And if you look at Trikafta, 200 nanograms in one, it's far better than Trikafta. This you can take once in a month. Trikafta you have to take 3 times a day. And if you go to day 2, you can see the 100 nanogram of reconstituted porosome, airway epithelium, it is able to overcome and is almost 50% better than Trikafta. Now on the lower panel on the left is an experimental in CFTR mice, nasal PD measurements. And you can see on the left part, the CF mice does not respond to forskolin, but if you add reconstituted with the porosome, you can see an increase, an immediate, and then the statistics on the right-hand side of this. Once we showed this data that FDA approved with giving us a orphan drug status on the CFTR treatment. So let's quickly, I want to keep time and I'm going to push this a little quickly. Similarly, in the brain of cadaveric patients, you see a drop of porosome proteins. Many of them are dropped, and some of them are given here. And you can see the 3 different proteins in the control on the left, the clear bar, and on the right, the shaded bar. And you can see those proteins are depressed in the Alzheimer's patient. Okay. And so the similar hypothesis now, what we could do is we can actually use the isolated porosomes from normal cells, brain cells, human brain cells, and use an organoid system, human brain organoid system that is developed, and then reconstitute it. And I will show you data on that and the reversal, but the way this can be taken up is through the nasal cavity, because to break through the blood-brain barrier, you don't need that. You can go through the olfactory lobe. If you take a little applicator and have it go through the nasal cavity, through the olfactory brainstem, the neurons that go to the brainstem for dementia and Alzheimer's get repopulated by the porosome, as I'll show you in a moment, and then that could be used as a treatment. But this will be maybe once in 6 months treatment, because membranes recycle in the neurons very slow in different tissues and differently. So this is a possible therapy that we are now going to complete in my studies and animal studies, and then it'll go to human trials. Let me share these data with you. So these are organoids. On the top, you see the control organoid, and you can actually measure them over time. This is measured over 130 days. And then you have AD organoids, brain organoids, and then finally the one with the treatment, without treatment. And if you measure these and do statistics, if you measure these, the top line that you see is control. It's growing very nicely. And the pink orange bar is the Alzheimer's growth, completely flat. It doesn't grow at all. The brain doesn't grow at all in Alzheimer's. But if you treat them with porosome and another flavonoid, apigenin, you can see the bar just below the green on top. You get a 50% protection or growth rate within 15 days of application. Very, very rapid. Now, this is just a gross morphology, yeah? Let me show you actually how do the cells look in the brain organoids inside. So the red is porosome proteins, as shown on to the left in control. You can see the blue are the DAPI or nuclear stain. And if you have Alzheimer's, you can see the porosomes are clustering tightly with each other, and there are huge gaps or dark spaces where the neurons have degenerated. Now you take those Alzheimer brain tissue and treat it with the therapy, reconstitute porosome and epigenin, you see again a redistribution. And that is shown below the distance between nuclei, the size of the nuclei, and so on, to give you some idea of growth and other things. So morphologically, you can see a reversal of Alzheimer's. This is the first time in the history of the disease that have shown a reversal. And now let me show you functionally. So Sony came up with a very brilliant idea of a brain on a chip where you can actually measure 3,000 to 4,000 firings of neurons in an organoid instead of patch clamp or one at a time. So we are not selecting anything. So the whole organoid are firing, and in fact, I will give you some idea about the amount of information that is generated. You can have a terabyte of information within 2 to 3 minutes. It's a huge amount of information that we can gather, and to show what happens when you treat and the organoids, which are AD organoids, and what is the rate of firing in AD versus control. And I'm going to show you that in the next slide. So on the left panel, there are 3 experiments ongoing. It's a screenshot. On the top is control, middle AD, and lower the reconstituted. But you pay attention to the bar. It's 100% in the control on the bar graph. It drops almost 40% of the control in terms of firing rate. And if you treat it, it is back normal. And then you can do a statistics on those. And in this one, you can see very clearly that you can reverse the disease functionally. So for the first time, both structurally and functionally, you can reverse the disease. So we were in New Delhi just a couple of days back, and there's Eli Lilly has just, India has just bought from Eli Lilly license of its treatment. What it does is decreases the amyloid plaque, which is important, but it doesn't treat the disease. It manages the disease. So what we are thinking is if you take out the plaques in early onset Alzheimer's, or even late onset, then you treat it with reconstituted porosome, you can actually not only reverse the disease, but cure it. And so it could be a combinatorial therapy. So we are very excited about this. We have been approached by several pharma companies we're working with closely. And it is exciting that for the first time, India has 8 million Alzheimer's patients, and there are many undocumented in this country. The US has 5. But more importantly, besides the patient losing themselves, it hurts the family. So you just multiply that by 5 or 4. And so that many people suffering. So for us, it's not about a drug that will make money or anything. It's about a cure that can change our lives. And I think that is very important, that's what we're pushing for. Okay, I'm not going to take much time because I can go on, but what I would like to do is just quickly tell you that animal studies show the same. You can actually reverse the disease on memory in mice, and this is the treatment. But I would like to quickly share with you in the next couple of minutes our goal at the Molecular Medicine Institute, in which we have many distinguished scholars. And what we are trying to do is put a focus on 3 important aspects to help society. One is, as I said earlier in my introduction, food security, health security, and energy security. Even though it's a Molecular Medicine Institute, others are taking care of the energy security, like the hydrogen you heard earlier. But food and security for health security is what we are for. We have several products on Porosome Therapeutics. We have many spin-offs in Boston from our company. I just spoke about 2 of them. So we are going to push and launch that, and areas that areas that do not have that also collaborate with globally, so that we can have others participate in the science that we do and train the next generation of scientists. Health security, we are very much interested. We have a spin-off called Nitroponics. We have a collaboration that we, in some regards, some aspects, capturing nitrogen from the atmosphere and capturing carbon, reduce carbon footprint, produce more sugar through our crops, and the use of nitrogenous fertilizer that protects the soil from erosion. So there are many projects that we're doing, and we welcome scientists in this community to reach out to us, and we'll be happy to take you in. So this is a Beverly Labs of MMI, Molecular Medicine Institute. And as I said, we are working on these 3 important projects. And with that, I will stop and thank you. Yeah.
Thank you, sir, for that enlightening lecture on porosomes. So, today we are extremely fortunate for getting this opportunity to listen to Professor Joachim Frank, one of the recipients of 2017 Nobel Prize in Chemistry. Professor Frank is a biophysicist best known for pioneering single-particle cryo-electron microscopy, a technique that allows scientists to determine high-resolution structures of biological molecules. Trained in physics in Germany, he developed key computational methods for 3D reconstruction early in his career and later advanced ribosome research while working at the Wadsworth Center and Columbia University. His revolutionary contributions to structural biology earned him the 2017 Nobel Prize in Chemistry alongside Jacques Dubochet and Richard Henderson. On behalf of all of you, I take great pride and joy in inviting Professor Frank to deliver the CSIR IMMT Institute Lecture of Eminence. Welcome, sir.
Director Narayan and members of this distinguished institute. It's a great pleasure for me to be here, and it's an extraordinary honor to be one of the speakers at the in a national lecture, and I very much appreciate this. I'd like to tell you about the revolution that cryo-electron microscopy has brought, and I'm referring to it as single particle cryo-EM because this is the particular contribution that I have made. I think I realized that this picture is sort of condensed. So a circle appears as an ellipse. I have no control over this. I see a different picture here, unfortunately. So today's medicine is guided by the knowledge of the way biomolecules are built and the way they act in concert in the cell. And with that knowledge, effective drugs can be developed, small molecules that are designed to bind to the target molecule and thereby change its behavior. And this, in a nutshell, is a definition of molecular medicine. And the knowledge of structure, dynamics, and organization is required on different scales. And these are associated with fields of structural biology and cell biology. And you have seen Banu Yena's depiction of a cell. And the message that I want to get across is that it's very crowded. Molecules are very sort of stuck together. They are difficult to visualize in this whole compact formation. So this really creates a rationale for in vitro studies. One wants to take the molecules out of the context and study them separately. And that's the way to get to really high resolution. And I just want to tell you about molecular machines in the cell. Molecular machines are everywhere in the cell. Molecules don't just bounce against each other and form some kind of random interactions, but rather they interact in concert in molecular machines. And so they are one that is particularly close to heart is the ribosome for me, because I put a lot of effort into looking at the structure. And in a case like this, we wish to know the structures of all components, but also the way they interact dynamically. And this makes it possible to produce targeted drug development to combat diseases. And so the important term here is reductionism. We take the molecules out of the context, and that's very unfortunate because we don't really know how they interact with one another in the cell. But at least we get all the way to atomic structure. And then we hope to be able to infer from that how they interact in the cell itself. Structural biology, I just learned, is a term that actually was coined by Don Caspar in 1972. Don Caspar is a really very eminent researcher who unfortunately died a few years ago. Structural biology is mainly focused on the structure of biomolecules in isolation. And this knowledge is of crucial importance in molecular medicine. We need near atomic resolution, 2 to 4 angstrom, to determine the position of every single atom in the protein. I'm sorry. Please. Okay. Structural determination is mainly done by X-ray crystallography or electron microscopy. I show you an example of a molecule, hemoglobin, very important. It's used in our blood to transport the oxygen. Here's a cryo-electron microscopy map, and next to it is an all-atoms map. All atoms map is very confusing. It shows all the atoms, but they are not connected with one another. One doesn't see the rhyme and reason for this. And that's why we use a so-called ribbon diagram, which shows the connectedness of the atoms, atom groups of amino acids along the chain. And this is actually how the proteins are formed. They are formed by a chain that then folds into an active protein. So structure determination has been done for a very long time, for decades, by X-ray crystallography. Molecules can be made to form crystals where they are arranged closely packed in exact regular order. And in that case, the atomic structure can be determined by clever methods of phasing. So the diagram on the right, these dots of the diffraction pattern really contain the information for recovering the structure. Here's Max Perutz and his student John Kenru with a model of hemoglobin. At the time, there was no better way of visualizing these when they had to carry these structures made out of metal or styrofoam around. And they received 1962 the Nobel Prize in Chemistry for determination of the structures of hemoglobin and myoglobin. And now this really begs the question, many molecules will not pack in regular order into crystals. They leave a huge gap in the knowledge of structures. And how about imaging molecules directly? You know, to look at molecules, can we use the light microscope? No, they're way too small, much smaller than the wavelength of visible light. Even the ribosome, a large molecular machine made from 52 proteins and 3 RNA molecules, will only show up as a blur. Here I have an actual juxtaposition of a human hair, the diameter of human hair. Next to it is a ribosome. This little dot there is the ribosome. So in terms of numbers, human hair is 80,000 nanometers, and the ribosome is 25 nanometers. So even though the ribosome is itself a very complicated structure made out of 52 proteins, it just shows up as a little dot next to human hair. You have to think about this idea, about the complexity of living systems. It's really unbelievable. And that's why electrons are used. If you compare the wavelength of light to electrons, the shortest wavelength of light is actually, I'm sorry, it's 400 nanometers, and this is half of it. For electrons, the resolution is less than 0.1 nanometer. So it's a huge change of resolution possible. I have the diagrams for image formation below each other, and you can see there is an exact equivalent in the formation of images. But for light, you have glass lenses, and for electrons, you have magnetic lenses. But otherwise, the paths of the rays are very equivalent. Now, a lot of people might be surprised how long your electron microscope has already been available. It was invented 1931, which is just a few years after it was discovered that electrons can be considered waves as well as particles. Ernst Ruska was the one who did this, and he got the Nobel Prize very late in 1986. And here's the diagram in his lab book where he sort of recorded this idea very meticulously. And So many years passed until electron microscopes were good enough and sophisticated enough to be able to be used for molecules. They were used for a very long time only in material science, and when they were used in biomedicine, they were just used for looking at tissues and so at lower resolution. So this is the idea of a transmission electron microscope. You have the electrons are generated on the top in a cathode, or it is also called electron gun, and then the anode is right below it, and the electrons are accelerated to the highest speed right below. And then a beam goes down the column. The column is under high vacuum, because otherwise electrons get stuck very quickly. And then you have an arrangement of lenses, and the upper lenses are so-called condenser lenses. They produce the illumination, and they focus the beam. And the beam then comes here. Actually, the object, the specimen is not shown. Where it shows specimen airlock, that's where the sample is situated. And underneath is the most important lens, the objective lens. The quality of that lens determines the whole quality of the image formation. And below the objective lens, an image is created, and that image is then magnified in 2 stages by the projector lenses. And right down there is the image. The image is formed, and what I show here is a sketch of an old microscope in which you could use binoculars to look at a screen, or the beam was recorded in plate cameras. And nowadays, there are simply digital cameras down here, and one doesn't sit at the microscope, one sits somewhere else. It might be the next room, and one has the image in front, and there is, of course, the whole digital storage and digital transmission. But what kind of image is formed? It's a so-called transmission microscope, which means that the image is formed from electrons that go through the object. So what we see is a projection in one particular direction. And what is the idea of a projection? Well, the closest to this is probably the idea of a jellyfish. We look through the jellyfish all the way, and depending on how much material is in between, you get a modulation of the intensity. Because otherwise, in the real world, we don't have things that we see through. We always see surfaces. But how do we get from these projections, to the three-dimensional structure? We need to have a particular mathematical operation. There is a scientist in Germany, Johannes Radon, who found out how the relationship is between projection and 3D structure, and how to recover the 3D structure. And this very important theorem has been the basis of doing three-dimensional reconstruction ever since. However, there were no computers available, so many years went by before it could actually be done. And the people who were the pioneers here are David Ressier and Aaron Klug. And unfortunately, only Aaron Klug got a Nobel Prize. David Ressier should have received it too. And this was a pioneering work, a three-dimensional reconstruction of bacteria phage tail using the Fourier-Bessel approach in 1968 at the MRC. And so I cannot go into it, but they used the theorem that I just showed you of this German mathematician. And but the curious thing is here is that they looked at a highly symmetric object. The individual molecules in the bacteriophage tail are in helical order. And so they were able to use a whole apparatus, mathematical apparatus, that made the recovery of the 3D structure very, very elegant and very fast. And so the so-called Fourier-Bessel function. But what I want to drive home here is a specific symmetry, highly ordered arrangement of the subunits, and a very specific mathematical way of reconstructing it. So now you change the symmetry, all of a sudden now you have to reinvent the software again, okay? Because you have to tailor the software to the new structure, which happens if you looked at spherical viruses with icosahedral symmetry or planar symmetry, two-dimensional crystals. So it was all very cumbersome because you had to find a way of arranging molecules in a highly symmetric way. And then you had to be a specialist in the use of a particular software to make it happen. So my mentor, Walter Hoppe, was one of the people who asked the question, why should we use crystals? Couldn't we just do it with asymmetric molecules? Simply, put them on the grid somewhere, and then tilt the grid in many increments, and then you reconstruct everything in the image field, along with the molecules that are sitting there. And the problem with that approach is that you accumulate electron exposure. And in this case, you get above 1,000 electrons per square. Electrons are highly damaging to the molecules. So if you get to something that goes more than 20 or 30 electrons per angstrom square, you're essentially destroying the molecule. So this way of this approach just doesn't work. Okay, and so I grew up in that environment there, and say, it's not going to work. It doesn't make any sense. You reconstruct something, and then in the end, it's like cinder. Everything is burned away. So I came up with the idea of the single particle technique. Instead of tilting the molecule, why not make use of the fact that they are randomly oriented in a sample to begin with? So they are already tilted. I don't have to tilt them. And instead of having one molecule, I have many, many instances of the same molecule, but in different orientations. So it becomes a completely different problem. But the beauty of it is that a very low exposure shot already gives us all the information that is needed. So the idea is structural information comes from images of single, which means unattached molecules that exist in many copies. Molecules are random oriented. Molecules are free to assume all naturally occurring conformations within limits. And a single snapshot may already give us hundreds of particle views, and as we collect more snapshots, more orientations will be covered until we have enough for reconstructing the molecule in 3 dimensions. Now, so it seems like it's a grand idea, it's a beautiful idea, but the problem is really in the details because there are no programs available at that time. And the most serious problem is that we don't know the angles of the molecules. So we don't know the structure, and we don't know the angles at which we look at it. So it's a chicken and egg problem. And so I had to be very creative here at that time and develop many programs and go very deeply into math, and until I had essentially accumulated a workbench of programs, 400 programs that were all sort of like bricks that we could put together in some kind of workbench. And so we gradually, in my group, we solved one problem after the other. One of them is, how to align images, how to classify them, sort them into different classes depending by appearance, find the projection angles, that was the biggest problem, and then reconstruct them in 3 dimensions, since programs didn't exist for general geometries at the time. Okay, so this is just an example for a proof of concept paper that I managed to get into Science in 1981 that showed, how beautiful it worked when we got images and cut them out and align them and then average them. We got very reproducible appearance with detail that had not been seen before. Okay, so then several years later, 1987, we finally got the first 3D reconstruction of a molecule, namely the large subunit of E. coli ribosome. But mind you, this was all with negatively stained samples. I have not mentioned ice before. We didn't have ice. The technique was not available. Okay, so I worked on the idea of the recovery of 3D information from projections of single particles. And other people worked on sample preparation. And so, and this was essentially the work by Jacques Dubochet, who got a Nobel Prize along with me. And I'm going to get to it. But I just want to drive home the fact in 1987, we had this beautiful 3D structure, first structure, which is here depicted in a light box with big sheets. It's a very big box like this. That was our way of showing 3D information. But it was not using embedding in ice. So I just wanted to, at this stage, I just want to make a demarcation and tell you about the stations of my career until that point, 1987. So I was first at University of Freiburg, then University of Munich, where I got my PhD eventually, then went 2 years to on a Harkness Fellowship to the US, then in the Cavendish lab in Cambridge. I was a group leader. Then I was at the Wadsworth Center as senior research scientist, and I became a SUNY Albany professor at the same time, a little later, while I was still at the Wadsworth Center. So here's Freiburg, study of physics. I had no idea I was going to wind up in biology. This is a so-called Vordiplom, which is an BS. And then I decided to go to Munich, and there I ran into electron microscopy just by accident. And I was curious what electron microscopy could do. That's how I found my mentor, Walter Hoppe. And by a curious coincidence, Walter Hoppe was an X-ray crystallographer. So with all the knowledge of that technique was there in the group, and all the knowledge of the literature. And he, for some reason, which I still don't understand, decided at one point in his career to switch into electron microscopy. So there was a group in which this kind of synthesis of ideas was already taking place when I joined. And so my whole horizon was formed by this whole X-ray crystallography background. And of course, what electron microscopy, I mean, the advanced EM has in common with X-ray is the Fourier space. We live in Fourier space, okay? We switch, we jump from Fourier space to real space wherever we can think most precisely. So here we are, University of Munich first, from a diploma part, and then Technical University from a PhD. And then, as I said before, I was for 2 years on a Harkness Fellowship in the United States, and it was very fortunate. I could pick any labs that I wanted, and I picked Cornell, and it didn't work out with Cornell because it didn't have a scanner, so I was redirected to the JPL. And JPL was unique at the time in it had the most advanced image processing facilities in the world. So I could scan images and reproduce images on film, which was essentially unheard of. And one particular feature was at the JPL, which is part of Caltech, by the way, was that they had an image processing system, and which still exists today, and it's called VICAR. And the VICAR system allowed them to process images that were sent down from the space probes, and they could do everything. They could stitch them together, make false color displays, statistics, and so forth. So I was able to use VICAR with my batch of programs. We simply plugged it in and could use the entire facility that they had. And that gave me the idea later for developing my own modular image processing system. So it was all came out for this accidental visit almost at the JPL. And then it was at UC Berkeley, and there happened to be Bob Glaser, who had all these ideas with low dose imaging of molecules, averaging, and so forth. And he even had ideas about cryo-EM, but he couldn't come up with the right kind of sample preparation method. So then, after a few months back in Germany, I went to Cambridge, and I was a group leader there for 2.5 years on soft money. But this was a fascinating experience. I was still in the old Cavendish laboratory and in Free School Lane, and this has a distinguished history. Ernest Rutherford was one of the directors, and he, of course, is associated with the discovery of the nucleus, and he was Cavendish professor in 1919. But the most spectacular thing here is that in the courtyard of this building, behind the Free School Lane, it was the former laboratory of molecular biology. It was a barrack in the courtyard. And that's where Max Perutz did his first X-ray experiments. And later, the lab of molecular biology, which was first part of Cavendish lab, moved out to the Addenbrooke's Hospital to become part of the MRC. Okay, so I could see the historical relics there. I don't know whether they still exist, but this was the picture at the time. And I already showed him with Ms. Kendrew. And then I went to Albany, New York for my first permanent position. And that's where I for the first time had the means to develop the idea, this fixed idea of single particle reconstruction in earnest. And that's how where I developed this spider system that I showed you before. And I've been for many years there in the Wadsworth Center, and then also associated with the State University in New York. So this is all the work for which I got my Nobel Prize later in 2017 was done right there in that institute and not at Columbia. So now that brings us to 1987. This was the addition of the cryo part of the single particle cryo-EM. And so what I always try to people to understand, we're talking about a confluence of two techniques at around 1987. This math software, but very inferior sample preparation, namely negative staining, single particles, and software without assumption of symmetries. And on the other axis here is sample preparation, vitreous ice embedding, which was applied mostly on crystalline samples and with software tailored to crystal symmetries. So these things merged together at a particular time, and all of a sudden there was an explosive development. With the plunge freezing that in and the creation of vitreous ice that Jacques Dubochet introduced, all of a sudden everything was possible. And so the idea is that a droplet of the liquid that contains many molecules in random order is put on the grid, and excess liquid is blotted away, and then the grid is very quickly plunged into liquid ethane, which is on liquid which is brought to on liquid nitrogen temperature, and if the plunge is fast enough, then the water doesn't have the chance to form crystals. It immediately produces goes into a different phase, which had not been known before, which is this vitreous phase, where it's like glass-like, and it has the same density as water at room temperature. and this is what it looks like. I don't have to explain. So these are the kinds of images that we see, but you notice that they look very flaky. These are images of the ribosome, look very flaky, very noisy. It's very difficult to discern any structure, and it's very clear that many molecules need to be averaged together before any clear image emerges. So we're not interpreting any single image. We always have to make statistical averages from many. But in terms of the progress of the entire technique, this was El Dorado all of a sudden. And it was incredible. All of a sudden we had a way of getting images, three-dimensional images of molecules that had never been seen before because they couldn't be crystallized. Okay? So the whole world was open. However, the resolution was rather poor. And there are several reasons for this. And one of them was that the microscopes were not developed far enough. They, wiggled around. Computers were very slow and very small. So it was a very tedious development until the manufacturers of electron microscopes really bid on this whole development. It was very, very courageous at the time. They invested a lot of money in it, And so everything improved. And at the time, when the first structure of the ribosome came out, which I showed here, right here, Nature in 1995, it was a very big deal for the Wadsworth Center. So they sent reporters in and made a big story out of it. And they produced this classical picture, where I look like a medieval scientist, bragging with my tools and stuff. So in 2000, we got the first really high-tech microscope. It was the first one in the United States of the Polaris series. And we made very rapid progress. And I told you about the ribosome so many times. I'm an accidental ribosomologist. I'm a physicist. I had no idea what I was getting into, and I adopted the ribosome as a beautiful demonstration and a model molecule with which to develop the technique. But I didn't know, without any biological background, what the hell it was doing. And then gradually it came to me what a central role it has in all life processes. So why the ribosome? Well, the ribosome has high contrast because of the high phosphorus contents, high RNA contents. It is large, which is good here for the technique, and it's stable. Okay, so these three features made it an ideal specimen. And then all of a sudden, I realized with our technology, we could solve problems, and that had really biological meaning. So the first problem was really to depict the whole elongation cycle of translation, how the molecules, dock to the ribosome, and then make it happen that then one amino acid per cycle is added to the protein chain. And so I'm showing an image here with Anders Liljas, taken several years later. He was the one who discovered this amazing mimicry between the two molecules. One is the ternary complex of tRNA, EFTU, and GTP, and the other one is EFG. So one molecules in the course of evolution emulated the appearance of the other one in order to dock at the same place as the ribosome. So after only a few years after this discovery was made, we were already able to depict in 3-dimensional the entire elongation cycle. And that was immediately adopted into textbooks everywhere. And then finally, New York, and that was my last station. I'm still there. Ribosome and time-resolved cryo-EM. So I went to some more advanced techniques. I'm going to just show you in a couple of slides later. So the incremental steps of the resolution revolution. Single particle cryo-EM, I think, is one part of it. Then electron microscope interfaced with the computer was a very big step because we could really do computer control and imaging on the same platform. And that facilitated automated data collection. They became much faster, more powerful computers. And then the discovery that with clever programs of 3D classification, we can extract multiple structures from a mixture in the sample, okay? So we don't have to highly purify one single molecule, but rather it is possible to detect the appearance of several molecules and reconstruct them separately from the same sample, from the same images. And then came the biggest milestone in the end was the huge improvement of image recording quality by a switch from film, which is very miserable as a recording medium, to electronic camera. And so the cameras became available in 2012 that enabled direct electron detection. This is a huge step in resolution and signal-to-noise ratio. And then all of a sudden, people talked about resolution revolution, and they realized that it's now possible to get resolutions that are on par with X-ray crystallography. So all of a sudden, there came the idea that we might even be better than X-ray crystallography, and obviously we're filling a very big gap in structure determination. But on top of it, our structures are really much more lifelike than X-ray crystallography shows, because in X-ray crystallography, molecules are essentially pressed in a straitjacket because it relies on the energy minimization of an entire crystal. And then was the development of the last generation of microscope. Here is the Titan Cryos from Thermo Fisher, and there is a similar, very large box by JOL. And these are the tops of the line, and they are now everywhere spread out. And the images are so much clearer than I showed before. So in the end, we wind up with a workflow which can be very much accelerated. And so this is essentially sort of a common workflow nowadays for structure determination. And in the end, when you have a high resolution three-dimensional image, from there you can infer the position of every single atom. Mostly you would rely on the knowledge of the sequence, but if we get really high resolution, then you don't need the sequence anymore. You can just read the signatures of the amino acids. And here is just to show you a single example of the very advanced image, three-dimensional image that we got after these cameras came out. This is the T. cruzi ribosome, which we imaged in 2016. When you see the false colors, they relate to the resolution. Very deep purple means the highest resolution at 2.3 angstroms. And at the periphery, because it's a single molecule, there's more flapping around, and the resolution goes more into 4 and 5. And nowadays, if you type in cryo-EM structures, you get pages and pages of structures, because everybody has now resorted to this. Many X-ray groups have stopped doing X-ray crystallography and are doing cryo-EM now. And so in the end, I wanted to show you 4 examples of cryo-EM advancing molecular medicine. And these are the human CDK activating kinase, and then the molecule that is implicated in cystic fibrosis, ryanodin receptor, which is implicated in severe, in several heart diseases, and COVID-19. So human CDK activating kinase, it's implicated in cancer in several ways, and the knowledge of the detailed atomic structure it will finally lead to a promising drug target development. And this here spells it out. It is involved in transcription initiation and the cell cycle of phosphorylating RNA polymerase II. Cystic fibrosis, we heard about this before from Banu Yena. These diseases of cystic fibrosis are due to numerous mutations, and we now know exactly where these mutations occur, and we can know how to target them. You see how many mutations are implicated in the disease. Any single one of them leads to the deterioration of the performance of the molecule. And then I was deeply touched by this email that I got a little later after I received my Nobel Prize. Dr. Frank, I want to add to the chorus of congratulations that you have no doubt received since Nobel was announced. I have a special reason to send you a thank you as well. My 7-year-old grandson, Jackson, was diagnosed with CF as a newborn in 2010. I've attached news that we have received this week regarding his 10 months on combination therapy. I just wanted to share this as a small expression of my gratitude. I was so touched because I did not associate my prize with some individual therapy result. But obviously, I have to take credit in some way, because down the line, all these discoveries will result in much more knowledge and much more choices in molecular medicine. And then this is the ryanodine receptor, and here it shows the one that is used by skeletal muscle, and there is another one that is very similar that is in the heart muscle. So this is it's a calcium release channel which is everywhere in all muscles, and there are dysfunctions which produce serious diseases like malignant hypothermia. And again, when we look at the location of mutations, we see that we can really track exactly where they occur, how they are related to the functional process, and so forth. And the knowledge, obviously, the knowledge of the sites of the mutations help tremendously in understanding drugs and also develop new drugs. And here is a whole list of diseases implicating mutations of the ryanodine receptor. And then COVID-19, and as you might have read, just 2 weeks after receiving the genome sequence of the virus from Chinese researchers, this team here at NIH had designed and produced samples of the stabilized spike protein. And it took 12 more days to reconstruct a 3D atomic scale map. This is all by cryo-EM, and submitted manuscript design. So it was published in March. That was the March of 2020, when all of a sudden the disease took over. So it was incredible that through the use of the technique, it was immediately possible to understand the mechanism by which the spike protein attaches to the host receptor. And so obviously there was a whole wave of studies everywhere, including at Columbia, and we have a very active group which takes samples directly from patients that have survived very serious illness, and then to look which antibodies were responsible for the recovery. And you can locate these antibodies, and then learn from that how you can design new mRNA vaccines. So there's a whole industry now behind it, and you will not be surprised after what I told you that the positions of structures in the database from X-ray crystallography has been rather stagnant over time, and whereas the cumulative number increases exponentially for cryo-EM. So very soon we're going to be probably at the same level as X-ray crystallography, which had 140,000 entries in the public data bank. We're probably going to have the same number very soon for cryo-EM. And so I just want to have a couple of slides that talks about what's the future, or what interesting things could be in the future. Well, some people say, well, we can press the resolution higher, but the resolution is already at 1 angstroms. This is already the best angstrom. So if we can get this to a rather routine performance, of course that would be great. But this is not really a very innovative way into the future. But my current interests are how to capture reaction intermediates and conformational changes. So now we talk about dynamics of a molecule. How do we get this from static images? Well, one of them is use microfluidics and start a reaction in the microfluidic chip and then stop it at a certain time and then look at reaction intermediates. This is, the time range is something like between 10 milliseconds and 1,000 milliseconds. The other one is, where I said state resolution. We can use hundreds of thousands or millions of particles, and then we get to a state where we can really actually follow very subtle conformational changes, and we see a continuum of conformational changes that are in a thermal equilibrium. And there is a way of recovering all this information and tie it together. So resolution in state space, we collect many snapshots and then make a deep data analysis. In the other case, we have to have special experimental equipment. So in conclusion, in recent decades, medicine has become very powerful by leveraging the knowledge of molecular structure. We speak of molecular medicine. X-ray crystallography has been the traditional technique to determine the structure of biomolecules, but many molecules do not form crystals, so the cure to many diseases was completely out of reach. Cryo-EM, which does not require crystals, is a new transformative technology for imaging biomolecules and determine their structure in their native states. And many biomolecules, such as ion channels and receptors, that are hard to crystallize for X-ray crystallographic studies, can now be imaged at high resolution. And based on the knowledge of the structure of biomolecules, drugs can now be developed that combat many diseases for which there was no cure. And this shows me with the group in 2018, just the summer after I was received the Nobel Prize. I still had a sizable group, and the group has now dwindled to 3 people. Okay. Thank you very much for your attention. Thank you.
Thank you very much, sir. We were privileged to hear your journey through the art of seeing the invisible. Now, Professor, I kindly agree to congratulate the scholar. Okay, so if you have one or two questions, please come forward. No, okay, you can move.
छोटा माइक दे दो कान में दर्द करेगा माइक नहीं
Good evening, sir. It is really a great pleasure and honor to listen directly the topic, the Nobel topic. Sir, actually, not now actually, after the discovery of cryo-electron microscopy and during the pandemic, actually, we have come across these molecules, SARS-CoV-2. So, as you said, actually, structure is very important to understand the structure and to diagnose the disease and also in discovering drugs. So when we are talking about any molecules, actually, we are extracting for our body, human body, for example, SARS-CoV-2 or any tumor molecules. So, for example, SARS-CoV-2. So when we are extracting for human body, that means they are at body temperature. So when we are actually freezing or transferring to cryogenic temperature and studying the structure, so is there any change or restructuring kind of things actually? How accurate is that actually? I am curious to know.
I understood very little. Are you talking about the change in structure is possible when it's being cooled?
Yes, sir. From body temperature, for example, some virus molecules, SARS-CoV-2 virus from body temperature to cryogenic temperatures.
Right. I mean, this is a completely general question, no matter what kind of molecule you have, right?
Okay.
Yeah. So it's been observed that the transition, when it's fast enough, essentially freezes the structure as it is at room temperature or whatever temperature you start off with. So obviously there is a small redistribution of energy levels and so forth, but essentially what experiments have shown, also MD simulations have shown, is that for rapid enough freeze, you will essentially get the same structure as you have at the normal temperature.
Okay, sir. Okay, sir. Thank you, sir.
Okay, so Professor, it was a wonderful lecture to listen to you. And my question is, after the availability of AlphaFold and Rosetta models, what do you think the trajectory of cryo-EM, that would be similarly used in the same amount, or it is going to be reduced, and how it would impact? What do you think?
Well, there is a common misunderstanding about the AlphaFold and what it can do and what it benefits. You have to understand that it relates on decades of experimental results. So it's a data mining of existing experimental results. So it cannot replace experimental results at all. And in fact, there is a whole universe of structures that have not been determined experimentally. And that universe is out of reach for AlphaFold. So it is very useful. It is very useful in essentially analyzing cryo-EM structures, as I just found out with my postdocs, because you have situations in which you have low resolution ligands attached to a large molecule, and then you want to essentially rule out many choices of structures. And if you do this by trial and error, it can take weeks. But AlphaFold made it possible to do this much faster.
Thank you.
So it's thrilling for me to listen to your lecture. So I want to ask you 2 points. One point is when you freeze the molecules, it becomes very brittle. The bonds are all vibration, rotational, everything is stopped in the bond. Because the protein, all the organic molecules, they are carbon, hydrogen, nitrogen or oxygen. So all the organic molecule freezes means they are very brittle. But in physics of point of view, when the electron is penetrating in that brittle structure, is there any speed and mechanical strength and that can damage the structure?
Oh yeah, I think this is, there are many parts that I cannot go into. Structure is really only produced by elastic interactions. Inelastic interactions, which involve transfer of energy, really destroy or change the atoms and produce ions, produce displacements of radicals and so forth. So fortunately, the elastic part is really the high resolution part. And so you can go about this in different ways. You can, for instance, use energy filtration, and then you can sort out all the inelastic scattering, and you only focus on elastic one. Okay.
Sir, it was wonderful lecture. Sir, you have given a wonderful lens to see the things, and if somebody has to see through your lens to get Nobel Prize, what are the remaining things which we should see to get Nobel Prize?
Well, I think the seeing part is very important, because when I look back through my career, I see many accidents, completely accidental things that I couldn't have planned. Okay, so there were very surprising discoveries, or I was approached by someone and that gave me a new idea. So all these things happened over time. And there is no recipe for this. But what I recognized that you have to see these chances. You have to see the opportunities. They're on the wayside. They're never on the clear path. You can never plan for it. And so I think this requires a way of going through life with a very open eye and what I always say, peripheral vision. You have to look at something in the periphery. It comes along and it's a total surprise. Don't discard it. You think maybe there's a message, okay? Maybe there's a center. And so that openness is very productive in terms of creativity.
Good evening, sir. Sir, this is a remarkable technology of the century, but every technology has its own limitations, like sample preparation and its cost, and regarding data collection and data interpretation. What you would say about it, sir?
Well, there are challenges on both sides. Sample preparation is a very big challenge. So it's you still have something like well-behaved proteins and badly behaved ones, and so you have to be very creative in sample preparation. There's nothing automatic there. And so this is a very big challenge to come up with a more streamlined way of solving this. And in the data processing, there's a danger because there's been the development of what I call black boxes, black box software. There is some kind of they take data and then they throw something out, and you don't know exactly. The user doesn't have control anymore. What's going on in between? He doesn't have an insight in all the very complex routines that are being used, and it's a market change from what existed there before. Before there were modular image processing systems like the one that I described, where you could really see the pathways. You can build pathways, and you knew the function of every single routine. You know it, and so you can be part of the whole discovery process, and that's no longer available. So there are new novices to the whole technique, and they might be very smart biochemists, and they know how to prepare the sample, but then in the end they fall victim to a black box, because they don't know how to operate that correctly, and then it could be many misinterpretations that come out of that.
Thank you so much, sir. Thank you, sir.
Good evening. We are really fortunate to be a part of this lecture. So my query will be, sir, India is the diabetic capital of the world, and most of, 1 in every 10 people are struggling with lifestyle diseases or regular intake of medicines to deal with it. And recently we got to know about this Ozempic shot. So I would like to know about the molecular medicine perspective of this Ozempic injection.
I missed it. Did you?
Sir, recently there was
So that's a weight loss drug. You're talking about GLP-1?
yes.
Yes. So GLP-1 is, it interacts in the brain neurons to make you not feel hungry, and so you don't eat. But the drawback for that is that when you stop the medication, you regain the fat, but you don't regain the muscle. So there is an ongoing program, and we have company spin-off that actually uses exosomes that will allow to protect the muscle and prevent them from loss when you are taking the medication. It's a Eli Lilly drug, of course, and so a lot of people are taking that, but you have to be careful.
Thank you.
So is that, so the question is, how is that related to cryo-EM? Is Professor Frank asking? Is it related to cryo-EM? Oh, she's talking about molecular medicine perspective. So in that perspective, I just answered your question. So as I said, you need to know the structure of a molecule, and cryo-EM is a very powerful tool to do that. With that question, I will also try to answer the AlphaFold business. So AlphaFold, as Professor Frank shared with you, there are limitations because it is designed to find new structures through the known structures in its memory. And so most of the structures in biology, especially for drug targets, is actually receptor or membrane associated, which are very difficult for any other technique except for cryo-EM. So the powerful tool for cryo-EM is crystallography. You can only get crystals of soluble proteins. Yeah? So membrane proteins are very difficult to crystallize, and therefore many membrane proteins have not been identified yet. That's what Professor Frank was explaining. Now, cryo-EM maybe will be able to solve that because cryo-EM can actually target membrane proteins, and once you know the atomic structure of membrane proteins, those new drug targets and new drugs can be developed.
I have a question. As we know that these kind of sophisticated instruments are very expensive for Indian perspective. So as you said that instead of direct view, you should have a peripheral view. Can you suggest that we can have some modifications into the instruments purchased with a high cost to experiment?
I can't hear anything. I can't understand it.
Sir, the sophisticated instruments like TEM and SCM, etc., are there in India, which are very expensive. Now we want to experiment on it by adding additional values to it. So, but there is a risk of failure, which may damage the instrument. So, how can you suggest that instead of just image processing, the condition of experimenting through different type of magnetic field or some kind of electron movement, we can add values so that it can have more resolution as well as more information can be brought out from that.
He is asking that in the whatever TEM and everything, it is expensive equipment, that how you have gone for the image processing, can it be improved further with the use of the new tools? यही है ना सवाल? The use of new tools like AI/ML in image processing, and only his fear that if he'll attach that one, then that instrument may become bad. So he don't want to experiment in cryo-TEM with the new techniques, but the people has a fear that how that instrument will work. That is a problem.
Yeah. Are you talking about the faceplate, or are you talking about contrast transfer function, or?
Sir, we want to experiment in designing a new TEM, provided we get the individual components instead of a big equipment, complete equipment. So is it possible to get it?
And
आप क्वेश्चन लिख के भेज देना. Right, yeah.
Sir, because in one of your steps, you said that you interfaced computer with the TEM. Then you got lot of images. There are lot of instruments which are not computer interfaced. Once we add computer to it, get the data, then we can get lot of new insights and maybe discover new things, which is great for the society. So for like electron microscope, there may be some other instruments, characterization equipment.
Hello, sir.
So just that answer. There is a new approach using faceplate is being developed in Berkeley, and you can get very high resolution, very clear pictures. But you have to use the computers. You have to use now faceplate that takes out aberrations and other things, makes it more clear. However, single particle is still the gold standard.
One last question, please.
Hello, sir. It was an absolute great lecture from both you professors. I have a question regarding diabetes only. So, in a case where a patient is both exocrine insufficient and endocrine insufficient, most of the treatments nowadays currently in India are based on drug and protein linked and cell receptor linked treatment mechanisms. But can we use a porosum reconstitution functional complexes to incorporate into the body and reconstitute the beta cells to have a time-based therapy or a cure?
Yeah, this is all a question to
Professor Jaina.
So one good example is we are working on type 1 diabetes. So what happens in type 1 diabetes, as you know, and it can be used for type 2 too, but in type 1 diabetes what happens is the body's immune system is now destroying the cells. Right? So your own immune
acting against the body,
destroying the cells, and you lose beta cells, and therefore you get diabetes. So what is the therapy? The therapy, new therapy is you take from skin cells, iPSC-derived stem cells. Through stem cells, just like the brain organoids and other things, iPSC-derived cells, you can make beta cells. And you can take those beta cells and transplant them into the right place in the body so that it is your own cells as not rejected, hopefully, but you still have to put it in encapsulation. Those iPSC-derived beta cells are only 25% effective. They are not fully transformed. Okay? They are not fully forming. They are only 50 to 60%, and therefore the potency and efficacy of glucose sensing and release of insulin is not 100%. That's one thing, and therefore what you have to do is you have to put a lot of those beta cells, which are derived from iPSC, and put them in the aorta or some other place, and that has a big problem because when you put those cells in a little cluster, the center cells do not get enough oxygen and nutrients, and therefore necrotized, die. Now what you could do is, the next step is to actually reconstitute the porosomes, and we have seen now in beta cells that we can reconstitute and get it 100% activated, and therefore instead of having 100 cells transplanted, you can have only 20 cells transplanted and you'll do the same job. So that's how you can overcome type 1 diabetes, and we're working on it.
Yes, sir. Recently in China, many studies have happened and the stem cell therapy is effective. Can we expect a similar step in India to treat the cause?
Well, sure, the technology is there, so if anyone is interested, we are doing it, and so we'll be happy to collaborate. So our Porosum Therapeutics does those type of work. It's being done in Boston in different labs. For example, Vertex Pharmaceutical is Doug Melton's lab, which was at Harvard. They actually have succeeded in doing these iPSC-derived beta cells, put them into capsules, and they're doing clinical trials. So if you're interested, yeah, you can.
Thank you, sir.
Okay, so with this, because you have lot of queries, you write down, we will pass it to Dr. Bhanu. So you can write to me that any questions you have, any queries you have, we will collect that and we will pass on to Dr. Bhanu sir that he will answer all your queries. Now, a big round of applause for both the speakers for the younger deliberation. Now I request our senior most colleague and our guru teacher, Professor O.N. Mohanty and Madam Mohanty to do the felicitation. I will also request Madam Frank and Madam Jena to join on a stage. And to make felicitation more grand, I request the Ganguly couple, the Hirendra Ghosh couple, Professor Biswal, Professor Kar to join on a stage and Dr. Thakur on a stage, so we will have a group picture during the felicitation. And Vino, you cannot be. Aye, karta bhai. This is Dr. Mohanty technology. Right sir. Right sir. भाई अरे आपको भी बोला है आप आ जाइए vote of thanks. No. Oh, you are right now.
Very good evening to all of you. Respected Chief Guest, Professor Joachim Frank, Professor Bhanu P. Jena, our Director, Dr. Ramanuj Narayan, esteemed dignitaries of the dais, dear colleagues, students, ladies and gentlemen. On behalf of CSR IMT, it is my honor and privilege to propose the vote of thanks on this momentous lecture of eminence. We are overwhelmingly grateful to Nobel laureate Professor Frank for your gracious presence and for delivering such an inspiring and enriching lecture. That was your pioneering contribution to science and your evocative interaction with our colleagues have truly made this occasion historic and memorable for the entire scientific fraternity present here. Our heartfelt appreciation goes to Professor Bhanu P. Jena for thought-provoking lecture on your pioneering work of Porosong. And also a big thank you for being instrumental in facilitating to bring Professor Frank to our institute. We sincerely thank our director, Dr. Ramanujan Nair, for his constant support and encouragement. We also express our sincere gratitude to all the dignitaries, including Professor Bose, Director Naizer, Professor Gangli, Director Aizer, Professor Kerr, and other invited guest participants for your gracious presence and enthusiastic participation in this event. We also thank all the media personnel for a valuable coverage of this event. A special note of appreciation goes to all my colleagues who have worked tirelessly to organize this event successfully, including Dr. Chauve, Dr. Manju Unnikrishnan, Dr. Manish, Dr. Nayak, and many more names are there. So with these words, I once again extend our heartfelt thanks to everyone present here. Thank you. Jai Hind.
I request all of you to kindly rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
This brings us to the end of this memorable event. Have a good evening. Jai Hind. Please join us for high tea outside and use this back door entrance. Thank you.